Hello, everyone. My name is Sara Rivera. I'm a master's student from Western Carolina University and this year's recipient of the Ross Taylor Bell and Joyce Rockenbach Bell Research Grant. I am here to present my ongoing thesis research focusing on the evolution of predatory fireflies, with emphasis on the firefly genus Vesta. So, I would like to welcome you to the Vesta Fiesta, where we are exploring the origins of predatory fireflies. Fireflies are a charismatic group of beetles in the family Lampyridae with tremendous cultural, scientific, and economic value. They inspire love and awe of nature, have given us important luminescent enzymes for biomedical research, and draw millions of ecotourists every year to firefly hotspots around the world. But despite the general love for these insects, they remain shockingly understudied, especially in tropical areas where fireflies are most abundant and diverse. One of the most interesting Lampyrid groups are the predatory fireflies in the subfamily Photorini. There are approximately 160 species in this subfamily spread across 3 genera, Photurus, Bisalonica, and Pyrogaster, and 2 subgenera, Diphoturus and Cephalophoturus. While all Lampyridae are predators as larvae, feeding on soft-bodied invertebrates such as snails and slugs, most lose the ability to eat as adults, all except for the predatory fireflies, which instead only eat other fireflies. Predation appears to be restricted to females of certain species, which are often referred to as femme fatale fireflies, and who carry a unique set of morphological characteristics. Their hunched bodies are more speedy and aerodynamic, and their long legs allow for prey capture in midair. To hunt, they often mimic the attractive flash patterns of other species to lure in males, which they then pierce with gnarly mandibles and chew using tooth-like projections of the clypeus. From their prey, they steal defensive chemicals, which they can then pass on to their own offspring to protect them from predators. This fascinating process imposes strong selective pressure on flashing fireflies and plays an important part in overall firefly ecology. However, we do not know how this system evolved. There are non-predatory members of the Photorini subfamily, which suggests a complicated history of many gains or losses of predation. Additionally, predation has only been recorded in some Photurus females, with only anecdotal evidence for predation in female Pyrogaster and Bisalonica. Predatory fireflies are in need of further study, and we have found a key that may help us understand how predation evolved. Introducing the special guest to our fiesta, the genus Vesta. Vesta is a taxonomically challenging firefly genus that has been found sister to or nested within the Photorini in both molecular and morphological phylogenies. This result was surprising because Vesta lacks many of the predatory traits seen in the predatory fireflies and is instead known for its elaborate pectinate antenna compared to the simple cylindrical antenna of the Photorini. Notably, these studies, while showing strong support for these relationships, may be affected by limiting sampling of Vesta, especially the neotropical species, and exclusion of associated taxa such as Diphoturus, a Photorini subgenus, and Delichnia, a sister group to Vesta. But that's where I come in, with hopes that expanded sampling of the Photorini, Vesta, and associated groups, combined with new morphological characteristics and field observations, could give us the answers we so desperately crave. Besides this uncertainty to its subfamily placement, Vesta is taxonomically challenging for other reasons as well. First, Vesta is a non-bioluminescent dark firefly known for its large serrate or flabellate antenna. While most well-known fireflies use flashing lanterns and large eyes to find mates, others use chemical signals detected by these large antennae. The antenna, eyes, and lanterns are traits used in sexual signaling and have traditionally formed the basis of firefly taxonomy. But recently, they've been shown to be highly variable and taxonomically uninformative. Reliance on these traits has resulted in messy classifications with overlapping diagnoses, which has happened to the Vesta. Taxonomic revision and the discovery of new, phylogenetically useful traits is needed so that further work can be done with the genus. Second, Vesta has a uniquely wide fracture distribution, with 10 species in South and Central America and 21 species in Southeast Asia, with no one in between. With such widely separated species, are these even a monophyletic group, or the same genus? How did they arrive at this strange distribution? Interestingly, there are no known Old World Photorini, which raises even more questions about the biogeographical origins of predatory fireflies. Third, if Vesta is part of the Photorini, What will that tell us about predatory evolution? If it is sister to the rest of the Fotorini, it may represent an ancestral pre-predatory state. If it is nested within the subfamily, it may be a formerly predatory lineage that may have secondarily lost predation. Either way, we may discover new exciting things about predatory evolution. To answer some of the questions about Vesta and the Fotorini and elucidate the origins of predatory fireflies, I have been working on building morphological and molecular phylogenies with expanded taxon sampling of the Fotorini, Vesta, and associated groups such as Delichnia, a Vesta sister group. I have had the honor of examining and borrowing specimens from multiple prominent collections, including the California Academy of Science, University of Georgia, Louisiana State University, Florida State University, Smithsonian Institute collections, and even the French and British Natural History Museums, where I also got to access type specimens. Everyone I have met at these collections has been incredibly knowledgeable and lovely, and I am eternally grateful for their help. With these collection visits, I was able to build distribution maps of the neotropical Vesta, shown here. These maps are divided not by political boundaries, but by biogeographical dominions. We can see that the Vesta are generally restricted to certain biogeographical areas, with distinct hotspots in Central America and northwestern South America. Considering their fractured distribution, perhaps Vesta initially colonized these areas from Southeast Asia, then diversified and spread out. Or maybe it's the opposite, that the Asian species first originated in these hotspots, then migrated to their current locations. Interestingly, we only see a single species, Vesta thorastica, crossing the Andes and spreading southeast into Brazil. Vesta is showing some interesting biogeographical trends, and considering Fotorini and Delichnia distributions as well would show some really cool, very useful patterns. When comparing the actual specimens, morphological similarities between the Vesta and the Fotorini are pronounced. The first similarity is the fusion between the frons and the clypeus. In most firefly groups, such as the Phytinus on the right, they are joined by a loose membrane, while in the Fotorini, Vesta, and Delichnia, they are fused. This fusion can be completely smooth or leave behind a hard suture. Either way, these fused plates are more rigid than those bound by membranes. This may be helpful for grinding up prey against the teeth on the clypeus, which is easier to do against a hard surface than a loose one. The second big similarity is the tibial spur pattern. The number of spurs on each leg is generally consistent among taxonomic groups. In this instance, the pattern of 0 spurs on the proleg, 2 on the mesoleg, and 2 on the metaleg is found across the Vesta and Fotorini, and is not a pattern yet seen in other firefly groups. Tibial spurs might be a valuable, phylogenetically informative trait we can apply moving forward, and even useful in dry specimens where other traits might be difficult to assess. The third set of traits we have observed is similarity in the ADA across the Fotorini, Delichnia, and New and Old World Vesta. These generally have a short phallobase, a long central phallus that tapers at the end, and parameres with long, membranous apical arms. Uniquely, the ejaculatory duct also appears to poke through the ventral plate of the phallus, as shown on the right. This is a novel trait seen across these taxa, but not seen in other firefly groups. The overall similarity among these Adiaga is striking, and definitely warrants further investigation. Besides gathering data from morphological matrix and tree, I am also gathering molecular data on these taxa. I am collaborating with the Stanger Hall Lab at University of Georgia to perform anchored hybrid enrichment sequencing on a variety of Fotorini and Vesta. This is the same technique used in the worldwide phylogeny of fireflies, so I will be able to compare my results to the larger picture of firefly evolution. So far, I have 12 new and old world Vesta, 4 Phyturus, 4 Bisalonica, and 3 Bioregaster, with more to be added from collaborators in Brazil. This data will go to building molecular phylogenies using Bayesian and maximum likelihood methods, which will be combined and compared with the morphological datasets to get a comprehensive view of the Vesta-Fotorini relationship. In addition to the work I have going, I plan to do fieldwork in Ecuador using a grant from the Coleopterous Society to collect more specimens. I visited Wild Sumaco Research Station in Napo Province this past summer, and I saw Vesta media, a species I do not yet have, as well as lots of Fotorini and even Diphyturus, which has not been included in any molecular phylogeny to date. Getting to include Diphoturus, Vesta media, and more Photorini will greatly enhance my project. I also hope to document predatory behaviors in the field, which can enhance the general knowledge of predatory fireflies. To conclude, using Vesta as a key to the Photorini will give us unparalleled insight into the incredible world of firefly predation. It's a dark world filled with sex, lies, violence, and beautifully deadly light shows. What more could you want from a research project? This project has built my collection, curatorial, taxonomic, molecular, and morphological skills. I even get to use my Spanish fluency in fieldwork in South and Central America. Understanding the predatory fireflies will help us better conserve these and other firefly groups before declines from habitat destruction, climate change, pesticides, and light pollution wipe them out forever. I am working for a world where future generations get to enjoy these beautiful beetles and the magic they bring. I would like to acknowledge Western Carolina University and the National Science Foundation for their support in my graduate education. I would also like to thank Yelena Pacheco and Dr. Catherine Stangerhall for their guidance in molecular testing for my project. Thank you also to the Coleopterist Society for their funding and their invitation to present, and an extra big thanks to Joyce Rockenbach Bell for honoring her husband, Ross Taylor Bell, and supporting my research. I hope I am making you both proud. Thank you so much for your time.